So thank you very much for the introduction, and if everybody would silence your cell phones, please. I forgot to do that for mine earlier. And we'll have time for questions at the end as well. As Jerry said, I'm the co-chair of the Spotlight on Science Committee, and on our behalf, we'd like to welcome you all to the first opening night of the Spotlight on Science series for this season. We're just one of the many outstanding programs brought to you by the Library Foundation of Newport Beach, which brings us together to hear from the world's leading authors, artists, journalists, doctors, and scientists on topics of interest, relevance, and importance to our community. The objective of the Spotlight on Science series is to transport you into the world of scientific research in an understandable and accessible way, and to describe the importance and inevitable impact of these endeavors on our lives and our communities. We'll be covering topics ranging from the infinitesimal to the infinite, exploring the sciences of biology, chemistry, physics, optics, neuroscience, electronics, the environment, and cosmology. Our 5 broad topical areas include the human body, our planet, energy and transportation, the worlds beyond Earth, and emerging technologies, and we'll be visiting these on a rotating basis each season. Last season, we learned about the role hydrogen plays in our California's clean energy future. We learned about how CRISPR technology is helping us unlock the genetic code. And finally, a perspective on California's tectonic activity and the unpredictability of earthquakes. You can find all of these in our prior season program videos on our website under the archives. Also, please calendar the upcoming series. We have one on quantum computing coming up on November 19th. And then next spring, we'll have the future of small satellites, followed by our grand finale, which is a talk on matter, antimatter, and the evolution of the universe, and will be a talk from a Nobel Prize winner, Barry Barish from Caltech. So it's an exciting season, so thank you for coming. Now, a bit more about the Library Foundation. Now, most of you know that the purpose of the foundation is to support discovery and connection beyond books. We are funding and organizing programs and embellishments to our library, including the other programs like the Woody Lecture Series, which showcases distinguished authors to spark community conversations, Library Live, which presents literary figures and emerging talent for adventurous readers, the Financial Literacy Workshops, which provide up-to-date information on managing your money and your estate, Medicine in Our Backyard, which highlights the latest in health and medical advances in association with our partners at UCLA Health, and the Book Discussion Group, which inspires people to open their minds and their hearts through reading inspiring and entertaining books through the year. The foundation also funds innovative library resources like the Media Lab, the self-checkout computers, the tech toys for kids, the Wayfinder, and the e-branch of digital resources. So really, we cannot present all these programs and fund these embellishments to the library without your support. So we do ask that we ask you to join or renew your membership to the Library Foundation, because it turns out the library itself is next to our public beaches here, the number 2 visited public space in Newport Beach. So there are over 1.2 million visits per year in person and through the website. So it's really a fantastic resource. And about our venue tonight, we'd like to acknowledge the Oasis Senior Center for hosting us. Our usual Friends Room at the library is closed due to construction, and it will be open, as Jerry said, and it reopened in February next year. It's a good thing that we're building that hall, because we wouldn't be able to fit this audience in that hall, because we're going to exceed probably 200 tonight. And speaking of Woody Hall, if you attended any of our programs last year, you've heard the fundraising pitch. And we did accept a challenge, the foundation, to raise half of the money for the new Woody Hall out of $24 million. It's going to be named in recognition of a founding gift by Bill Whitty and Keiko Sakamoto, and it will actually be a community-wide asset and a city facility, and will support not only the library, but literary, cultural, artistic, educational, and even business events for years to come. So while we've raised over $11 million on our way to half of the donation, we do urge you to join us in making this vision a reality, and to help us bring the hall in on time in March next year. So let's talk a little bit about tonight. Our talk tonight is entitled From Molecules to Mankind, Tracing Life's Origins. Our speaker is Dr. Irene Chen, Professor of Chemical and Biomolecular Engineering at the UCLA Samueli School of Engineering. She received her BA in Chemistry and an MD PhD in Biophysics from Harvard, and then taught at UC Santa Barbara before joining as professor at UCLA in 2020, where she teaches courses on the scientific origins of life, and her lab runs projects on understanding both the beginnings of life and antimicrobial resistance in infectious disease. She's a recipient of the Searle Scholar Award, the NIH New Innovator Award, and the David White Award for Outstanding Contribution in Astrobiology. She was a Simmons Investigator for the Collaboration on the Origin of Life from 2013 to 2023, and presently, after she wraps up her day teaching courses and leading research, she heads back to campus where she is an Olympic Hall faculty in residence, which means living 24/7 with 1,000 students, as well as her young family. I think there must be an award for that too. So please join me in welcoming Irene to the Newport Beach Public Library.
All right, thank you so much, Randy, for that introduction, and thank you to the foundation and Bruce and Randy for their invitation here. Okay, let me just. Okay, so this is a very ambitious title. Honestly, I did not write this title. I think Kunga wrote this title, but it probably oversells things a little bit, but it's great to bring you guys in here. What I hope to do is to make this question about how life began, how we got from molecules to mankind. What I hope to do is to make that a little bit more concrete and to explain how we think about this from a scientific engineering perspective. Okay, but to kind of summarize our current understanding of all the stages that had to take place in order to get from molecules to us, this is summarized in this mural, which is hanging in the NASA Ames Research Center. And this depicts the process of the origin of life. So starting, okay, let me get up my pointer here. Starting up here, we have the cosmos and the Big Bang producing all this matter. And at some point, we have planets that are gravitationally attracted matter. And then the Earth, which condenses and then cools to the extent that we can get liquid water. So once we have our Earth starting out kind of hot, but then cooling over time, maybe 4.5 billion years ago, we have liquid water and some very simple molecules like methane, just a few atoms hanging together in small molecules. And the thinking is that we have these small molecules to start with, and over time they can collide with each other, form bonds, chemical bonds, make slightly larger molecules. And then maybe some of these molecules have special kind of repeating properties that can turn into polymers, chains of these molecules. And these chains are things like proteins, which you've probably heard about, proteins, peptides, ribonucleic acid, RNA. I've kind of written it down here because it's going to play a large role in what I talk about. DNA is also one of these types of molecules. And then once we have these kind of larger molecules, then they self-assemble. There must be some Darwinian selection, some natural selection also for the self-assembly that creates very primitive cells. And then once we have evolving cells, then we can get to multicellular organisms once they're communicating with each other, sticking together, helping each other, building communities, and up into larger and more complex organisms. And of course, because this is NASA, it all ends with astronomers looking up at the heavens. So what are the details of this process? How do we even study it? This is something that took place, billions of years ago. So how do we think about this? Now I'm going to kind of move over to some inspiration from science fiction. So Star Trek: The Next Generation, Captain Picard over here. In this episode, Captain Picard actually meets his old Starfleet professor, who tells him that he's discovered some kind of ancient code in some archaeological findings. And a lot of hijinks ensue, and at the end of this season, they end up, and a bunch of other species, humanoid species, end up on this isolated, uninhabited planet. And they discover a recording, which was produced by this individual, who says that all of the humanoid species that we see on Star Trek are related because they're derived from this ancient DNA that this species, this original species, had spread. So I think this actually illustrates really nicely one approach that you could take to studying how life began, which is that you start from looking at the organisms we have, and then you try to go backwards in time and infer some kind of earlier organism, some simpler version. So this is one philosophy, you could say, one like approach, almost like a historical perspective of studying the origin of life. You study the existing life and then look back. And on our diagram, that would kind of mean studying life and looking for simpler forms, maybe microbial forms. They're simpler, there are fewer genes, and trying to go back as far as you can go. And people will call this this sort of limit of how far you can go the last universal common ancestor. Like, what was that microbe that gave rise to all the life that we have? And we can make some inferences about that going in that direction. Okay, so that's one approach, but it doesn't get us really all the way to the beginning, because you could say there's a lot of mysteries here. You know, how do these molecules assemble? What is driving them forward in this kind of timeline of complexity? So to think about that, we can look at this episode of Star Trek, where this is the season finale, where Captain Picard is kidnapped by the alien Q. So Q is omnipotent, he can travel anywhere in time and space, and he takes Captain Picard back to the primordial Earth. So this is the primordial Earth, I hope you can see that. And there's some liquid water there, so they look down. There's this puddle of greenish water, greenish gooish water, and Q stirs his hand around and he says, this is where life started. There's some small molecules in here, amino acids, they're all gonna bump into each other and life is gonna start. So this is kind of the second philosophy that we have to understand the origin of life. It's starting from some chemistry, like putting in some simple molecules that we can all agree were prebiotically plausible. You know, maybe we find them in stardust or we find them in meteorites. We think they're plausible as being made without biology. And then we ask, can we build up from there? Can we figure out ways, can we, as you know, chemists or biochemists, figure out ways to put these molecules together in a timeline that would lead to something more complex like a cell? Okay, so rewinding time to the 1960s, this idea of trying to make life has been there. So here's one recipe that was written. If you press a piece of underwear soiled with sweat together with some wheat in an open mouth jar, after about 21 days, the odor changes, and the ferment coming out of the underwear changes the wheat into mice. So we probably also, many of us experience things like this in our lives. So this recipe was given by a chemist. This guy here was actually, a serious chemist. He's the first person to realize that there were distinct gases in the air. Here's his shot at, creating life. So let's see, what can we do about this? What are the realistic ingredients we can put in? Okay, so back on our timeline here, we're trying to go forward in time from these really simple ingredients, trying to kind of meet in the middle with this other approach coming backwards from life. And I'll be talking a lot about RNA as being a critical molecule that is central for its ability to evolve. So let me just talk about RNA for 1 second here. And if you're asking, how do you get RNA? I will come back to that. So let me just describe what it is so that we can see what the challenges are. Okay, so RNA is ribonucleic acid. And it's very similar to DNA, which maybe you've heard, deoxyribonucleic acid, which is the stuff that makes genes. And ribonucleic acid is often thought of as kind of an intermediary between the genes, which are DNA, made out of DNA, and proteins, which is basically everything that does anything in your body. So we have our DNA, which can self-replicate. There are proteins that help it replicate and copy that information. Maybe sometimes you hear like the DNA or your genes or the instruction manual for the cell. So that's the information in the DNA. And then to actually make the proteins out of the DNA, your cells go through this intermediate molecule, RNA. And RNA is a chemical cousin of DNA. You could say it's very similar chemically, just like one atom different, basically, between DNA and RNA. And it travels out of the nucleus, out of the genome, where DNA is kind of holding the information. RNA takes that information out and allows the cell to turn that information into proteins. So this process of taking DNA information and turning it into RNA information is called transcription. And this process of taking RNA information and turning it into proteins is called translation. Okay, so RNA is kind of right now in our cells a little bit of a middleman, and we often think of RNA in this kind of limited context, and the first-year biology students will mostly think about RNA in this context. But actually, our understanding of RNA has changed quite a bit over time. So what I just told you is how we thought of RNA in the 1960s. So life is based on DNA, that's where all our genes are. DNA uses RNA to make proteins, and the proteins are the things that are doing things. They're catalyzing reactions, they make up your fibers, et cetera. Then in the 1980s, people discovered, oh, actually RNA can do some things by itself. It can also catalyze reactions. It can cause reactions to go faster. And this discovery was a Nobel Prize-winning discovery that RNA could make reactions go faster. And we'll come back to this catalytic RNA. And then, in the 2000s, people started discovering a whole zoo of different kinds of RNA. So there's microRNA, there's silencing RNA, there's long non-coding RNA. There was a lot of RNA that people had previously not noticed in cells. And there's some Nobel Prizes in here too. And then, nowadays, the 2020s, life is a seething mass of RNA, sometimes uses DNA to take notes. What do the proteins do? Errands for RNA. So you could say we're still living in this world that's dominated by RNA. Okay, so let's now, with this understanding, kind of go back to our RNA. We have RNA, you can think of it as a sequence. This is that instruction manual aspect where RNA has an alphabet of 4 letters, and you can think of this kind of alphabet as you can think of RNA in terms of that alphabet and the information that's in the sequence. Then you have the structure of the RNA, which is very similar to DNA. You can kind of see the similarities to that double helix. And then you have some RNAs that can fold into very special structures that are so complex you almost they make you think of proteins. So this is a catalytic RNA where you can recognize little double-stranded helix here, but you can also see this sort of complexity of the fold that allows it to grab onto other molecules and push them together, create the right environment for reactions. Okay, so we have RNA. It can be a genome, it can be an information carrier, and we see this in viruses, like the influenza virus is an RNA virus, coronavirus is an RNA virus, and then we can also have functional molecules that are made out of RNA. So just to, it's easy to say those words, but I also just like to think sometimes about how amazing it is that RNA has this property. So if you think about RNA as the instruction manual, just the words, think of what it would be like if the words that were in your book could actually fold up into the thing that they were describing. So if you had J.R.R. Tolkien's passage about Shadowfax, those letters, those words could actually fold into a horse. That is what RNA is like. Okay, so the RNA world theory goes back to the 60s by Francis Crick, who many of you might recognize him as one of the discoverers of the structure of DNA, Leslie Orgel and Carl Woese. They had this idea based on the fact that RNA could fold into complex structures, that RNA could have been the foundation of life, because instead of having DNA and RNA and proteins, maybe you could just have RNA, and RNA was doing all of the jobs that nowadays we need 3 types of molecules to do. So this simplification would make life easier to create. And so there's this theory that there's this intermediate stage of life that was based on RNA. And there is some evidence from biology for this idea. One of them is the centrality of RNA in metabolism. So when I say that, I mean there are some really special molecules in cellular metabolism that look a lot like RNA. And ATP, if you've ever heard of that molecule, that's kind of the molecule that carries energy in your cell. You could think of each ATP molecule as like a little battery that stores energy, and when you break a bond in the ATP molecule, it releases that energy. That is so critical to life, and ATP is the A in the alphabet of RNA. So there are many other examples of these kind of central metabolites that are so important in biology that seem to be RNA. And then we have these RNA catalysts, the fact that RNA can be catalytic as a piece of evidence there. And then in around 2000, what people thought of as the smoking gun of the RNA world was discovered, and that was embedded in the structure of a biomolecular machine called the ribosome. So let me just describe that because I think it's a really interesting finding. We don't have any way to really, find a fossil of the RNA world. It's been so many billions of years that that kind of preservation is impossible. But we can look for this kind of circumstantial evidence, the smoking gun type of evidence. No direct evidence, but maybe some circumstantial evidence. So what we, as a field, as a community, call the smoking gun of the RNA world is the structure of the ribosome. So the ribosome is the complex in your body that turns RNA information, translates RNA information into proteins. So this is a critical task in taking that DNA, those genes, and making products from it. So what was discovered is that the ribosome is made out of RNA and proteins. And when that was known for a while, and people assumed that the protein was the important part of the ribosome because proteins are the important part of almost everything. But when the structure was solved, it was seen that everything in here that's brown is RNA, and the proteins are this purple. And what you see is that the center of this structure, where all the chemistry is happening, that is made out of RNA. So the ribosome, the machine that makes proteins, is actually an RNA machine. So if we take these two things, the ribosome makes proteins, and the ribosome is mostly RNA, and the critical parts of the ribosome are RNA. This tells us that RNA, even today in our bodies, is making proteins. And that's thought to be like the smoking gun of the RNA world. Why would you have an RNA complex making proteins unless it had some historical reason? Because proteins do everything else in our bodies. Okay. So this is kind of what we're going for here for making a simple type of life. I remember going forwards in time from those chemicals, from chemistry, and transitioning into something like a simple cell would be this RNA-only metabolism. So RNA that can replicate itself, and RNA that, and some of those special RNAs that can catalyze reactions and kind of take the jobs that proteins do now. Okay, so we've kind of danced around this question, what is life? Is there a definition for life? And there are so many papers written about this, and it's a huge topic, not only in science, but more broadly. But what we as kind of synthetic biologists, if you can call it kind of a way of trying to make biology, what people have adopted in this field is a definition that was put forward by Gerald Joyce at the Salk Institute, and in a working group that NASA held in, I think, the 1980s or so. He wrote down this definition of life as a self-sustaining chemical system capable of Darwinian evolution. So what's really nice to me about this is that it doesn't tell us anything about what the life is made out of, other than that it's made out of chemicals, which distinguishes it from artificial life, like computer viruses or something like that. So it has to be made out of chemicals. But it doesn't say anything else about what the life does, except that it has to be able to evolve. And this property, we can agree, is critical to life, and who knows, maybe it's the only thing that's critical, but many of the other definitions of life can kind of be encompassed by this idea of evolution. So now our question is, maybe is all you need RNA? So let's see if we can get to RNA and making this type of system. Okay, so what are we starting with, and what can we get kind of in the very earliest stages? Around the same time that the structure of DNA was being solved, this is the 50s and 60s, Stanley Miller, who was at UC San Diego for his career, and Harold Urey at the University of Chicago, did an experiment where they mixed together very simple molecules. So these are, just a few atoms in these molecules, some water, some of these reduced gases, and added a spark, electrical spark, to simulate lightning as a kind of input of energy. And they found that this combination could make simple but still interesting biomolecules like amino acids. So amino acids are the building blocks that, if you stitch them together, will make proteins. So finding amino acids that could make proteins in the output of this experiment showed that you could make these building blocks of life from a process that had nothing to do with life. And this is one of the apparatuses that Miller built back then. Here's the reaction chamber. Here's where the spark is. The gases kind of go up here and react, and you have your water down here. And other evidence kind of came in kind of around the same time that you could make biomolecules like adenine. So adenine is the A in RNA and DNA, and you could make those from cyanide. And if you look at the cyanide molecule and stitch a bunch of them together and form some rings here, you could get adenine. So nowadays, maybe you've heard about the recent news from the OSIRIS-REx mission. You can ask the same question, can we get these same biomolecules? You're telling me that maybe abiotic processes can make these amino acids and simple molecules. Can you also find evidence of that? And we can look at the OSIRIS-REx sample return from an asteroid and say, do we have those molecules in there? And they have indeed found many of these molecules. And there have been samples taken from Mars, kind of left on Mars by the rovers, and there's a hope that NASA will someday return to Mars and pick up those samples and return them to Earth. So this is an ongoing kind of field of inquiry of what can we find out there. Okay, so let's say, we can make those amino acids, those simple molecules. How about the building blocks for RNA specifically? So one gentleman asked me earlier, this seems to have been an old problem that was not solved for a long time. And I would say, yes, there was a period of time when people did not think that RNA was prebiotically plausible. But in the 2000s, this synthesis was published, and it just kind of shows you that our knowledge is limited. Just because we've tried really hard and it didn't work doesn't mean that it can't work with the right approach. So Finally, in the 2000s, there was this kind of chemical breakthrough where people realized how to make a nucleotide. And you don't have to really understand exactly what this is, except to say that this is a complex molecule, and if you put together many of these molecules, this is a nucleotide. If you put together many of these nucleotides, then you can make RNA. And you can get there from starting from these very simple molecules, just a few atoms, these simple prebiotically plausible atoms. Okay, so we think we can make RNA mononucleotides. Can we make them into strings of polymers? And it turns out there are a lot of ways to do this. One of them is what's illustrated here in sheets of clay. So clay minerals have these kind of like spaces in them, these layers, and it turns out that some clay minerals have the property that if you add nucleotides and a source of energy to these clay surfaces, then the nucleotides will line up and polymerize on these surfaces. So this is one way to do it, but there are other ways to do it. This polymerization process can make our longer strands of RNA. Okay, next. So we have a long strand of RNA. Can you make a copy of it? Because you need to self-replicate. Can you copy this RNA? And again, the answer is yes. There's been a lot of work in this area for the past few decades. This is maybe I won't go into the details here, but just trust me if I say, like, this is a small piece of RNA, and this is a long piece of RNA, and these are kind of the intermediate stages of making that long piece of RNA. And you can do that without any enzymes in there, so this is totally a chemical system with no biological parts in there. So we can make them mononucleotides, so we can make those small RNA building blocks, we can polymerize them, we can make them into long chains, and then we can copy the information in those polymers. Okay, so what's next? Can you make self-replicating RNAs? So here's one system where you can take two RNAs, and they're just labeled A and B. And if you have a template in there, so if you have a longer version of these two molecules, then the fact that you have that template will allow your other two molecules, A and B, to self-assemble and create a covalent bond. So you can go from taking kind of two pieces of RNA and stitching them together to make a longer piece of RNA. And this is one kind of self-replication. Maybe a more general type of self-replication is if you had an RNA catalyst, so an RNA that could grab mononucleotides and a template sequence and copy that template using the mononucleotides. So this is kind of like it's been called the RNA xeroxase. So it's xeroxing a strand of RNA information. And this exists. People are still working on making it better, more efficient, be able to copy more information, but I think we're on the road here. Okay, so sometimes people will bring up this question, and now that we have a system to work on, which is RNA and these kind of catalytic RNAs, now that we have a system to work on, we can start to address some of these fundamental questions that people have. So one question is a chicken and egg paradox that was first brought up by Manfred Eigen, a Nobel laureate many years ago. And he said, okay, the first ribozymes, meaning the first catalytic RNAs, they must have replicated by chemistry alone, because there was no biology around, so it must have all happened chemically. But chemical replication is very error-prone. Nowadays, our bodies, our cells, have many mechanisms to ensure that the copying of a sequence is done with high fidelity. It's done very accurately. But back then, none of those mechanisms existed, so how could you get a system to propagate information if you had so many errors being introduced? And if you have too many errors being introduced by your chemical process, then this leads to something called the error catastrophe, where the information is lost. So let me just illustrate this here. This is the name of a lake in western Massachusetts. I'm not even going to try to say it. It's a Native American word. Apparently it means you fish on your side, I fish on my side, nobody fishes in the middle, which is kind of distressingly resonant right now in our times. But this is clearly a very long word, so let's see how good we are at replicating this word. So this is another sign, clearly referring to the same lake, and it is identical except for 2 errors. And I honestly don't know which sign has the errors. But this shows you that replicating a long string of information at even a relatively low error rate, let's say, I don't know, 5–10% errors, still, if you've got a lot of information to replicate, you're going to have mistakes. So if you made a copy of this and then tried to make a copy of that, and et cetera, then you can see how the information would get lost. And we can see this in modern day organisms. So if we look at how much information there is in a genome, so the genome size relative to the mutation rate of that organism, we can see this downward trend. So for example, humans, we're up here, we have a very large genome, lots of information, and at the same time, a very low error rate. So, 1 in 10 to the 10, 1 in 10 billion errors per replication. And then, as you go up here, and viruses are kind of where you start to lose a lot of this accuracy, you've got shorter genomes and high error rates with viruses. Okay, so I won't go into all the details of how we solve this problem, but just to say this is the type of problem that we have, and it turns out that the solution is in the chemistry. And what's really nice to me about this is that you don't add any mechanisms here. What happens is if you've got a strand of RNA and you're copying it, let's say there's an accident and an inaccuracy, you get an error in there. It turns out that finishing this strand is very slow. So that fact, the fact that you can make these perfect copies fast, and then just the fact that you have an error slows down the whole process, that is kind of like an intrinsic error correction. And you didn't have to introduce any mechanisms outside of what was already in the chemistry. Okay. Another question that I think often comes up is, what's the role of chance in the origin of life? If we had another Earth and we started that Earth again with an RNA world, we seeded it just the same way, would we get humans again? Would we get our central dogma, our DNA, RNA, protein system, would we get that again? And Stephen Jay Gould put this very nicely in terms of how we would try to test this question. Suppose we could rewind, or maybe start experimentally, many different replicates of the same experiment, and then repeat the evolution. So we'll let the tape run again, see if the repetition looks at all like the original. And then, if it does, then we could say, perhaps this is something where chance was not very important, or maybe this is something where, it was a flip of a coin. So we can address this kind of question in the lab, and I'll just say briefly that the way you can do this is to set up many replicates of an evolutionary experiment, let it go under certain conditions, try to keep everything the same except for the fact that you have multiple test tubes doing this, and ask, am I going to get the same results? Do I get the same sequences, the same genome, same RNA sequences out of this test tube evolution experiment? And if you do that, it turns out, at least for some of the systems we've looked at, it turns out that you get quite different solutions. So in this system, in the RNA world, depending on where you start in that RNA world, you might end up in a very different place. And I like to kind of make the analogy to a couple of different geographical structures. For example, forests of Finland, apparently very interconnected. You can imagine this is a space where no matter where you start out, suppose you're trying to find like the center of this system. No matter where you start out, there is a pathway to get there. But if you're in a system with more isolated islands, suppose we have a high point over here. You start out over here, there's just no way for you to traverse this type of landscape to get to the highest point. So this is saying that there is a role for chance in here, and maybe depending on how you set up your experiments and what you're looking at, perhaps the role of chance might be higher or lower. Just want to, I know that was a lot of maybe chemistry and experiments, and so I just wanted to take a step back and think about this cartoon from Randall Munro, where he kind of classifies science into three types of research. So one is we apply a standard theory to novel circumstances and get some surprising results. And two is we apply a novel theory to standard circumstances and get some intriguing results. And then we have this guy here, finally a map of every tree. And sometimes I feel like the science in the origin of life, a lot of it, we do our best, to be as, kind of, straightforward and like hypothesis driven as possible. But sometimes you just go out and, try to map the trees. You just see what's in that sample from the asteroid. You just ask, if I do this experiment 100 times, do I get different results? Okay, so I sympathize with that third guy a lot. Okay, so that's RNA. So let me just throw in a little bit more complexity, because you're going to say, Irene, you said molecules to mankind. We only got to molecules. So what's next? Can we get to a cell? So that's kind of the next level of complexity here. So here we take kind of inspiration from Albert Einstein, who said everything should be made as simple as possible, but not simpler. And what I think is actually hilarious about this is what he actually wrote, which has been distilled here. What he actually wrote was this very long explanation of what he was trying to say. So it's been simplified into this quotation here. Okay, so is RNA enough to make a system, or is that too simple? So many of us in the field think that RNA by itself is actually too simple. And here's a couple of reasons. So one reason is that if we have a system that has RNA in it, we have to be able to get food into the RNA and waste out from the RNA. And if you're in like a free solution, then there's no distinction between where the RNA lives and the rest of the media. So the way to keep your RNA kind of protected and away from molecules that could degrade it, or to let it interact with the outside, is to put it inside a cell. So this is one reason. A second reason might be a little bit less intuitive, but let me try to explain. So if you imagine a free solution and you have many of these RNAs that are Xerox machines. So this RNA can go around and pick up another RNA and make a copy of it. If you have this kind of like amazing kind of RNA with this property, it's going to spend most of its time in a solution. If it's able to interact with all of the RNAs, it's going to spend most of its time copying other sequences. So the Xerox machine makes a copy of something, picks up another RNA, makes a copy of that RNA. It never makes a copy of itself, which is what you want. You want it to you want it to self-replicate. So because of this property, not being able to copy itself, but only making copies of other molecules, in free solution, this Xerox machine does not increase. And actually, you could say it gets parasitized by all of the other RNAs in the solution. So there are a lot of solutions to this kind of problem. It's kind of a cooperator-competitor problem. One of the ways to enable the RNA, a special RNA like this, a altruistic RNA, if you will, to survive, is to put it in a compartment where it only interacts with a limited number of molecules. And this kind of compartment will keep it together in a kind of cooperative cycle. Okay. So we think that the next thing that we need, not just RNA, probably arising in parallel to the RNA, are these artificial cells. or I should say, now that we make them in the lab, they're artificial, but back then they would have been naturally occurring cells. So what can we use to make these cell membranes out of? What do you need to make this cell? So fatty acids are probably familiar to you as the main component of like olive oil. Fatty acids are this kind of soapy-like molecule. They have this fatty region, and they have a region of them that likes to interact with water. So they have this water-loving region, and they have this a water-fearing region, and that's what makes them self-assemble into structures where all of the water-loving parts of the molecules are on the outside, and all the water-fearing parts of the molecule are on the inside. It gives you this nice kind of membrane, cell membrane. Okay, so where can you find these fatty acids? You can find them abiotically in syntheses that simulate hot springs or hydrothermal vents. These will produce fatty acids. And we can find them on meteorites. So this is kind of like the proof that these fatty acids could be made abiotically, is that they can be found in these meteorites. And if you take a sample from a meteorite and you extract the organics from it, and you resuspend it in water, it turns out you can make these structures that look a lot like cells. So these are, there's nothing living in here. This is just extract from meteorites, and it's just showing you that if you add water, you get these self-assembled structures from fatty acids and other similar molecules that are already there. And they interact, they play nicely with RNA. So here's a system where the RNA is labeled in red and the fatty acids are labeled in green, and you can get these RNAs encapsulated inside. Okay, let me just skip that. I think another really interesting thing that we've been learning is that the membrane is not just a passive container for the RNA. The fact that you have RNA or any other molecule inside a membrane, inside a container, the fact that it's in a container changes its properties. And if you look into a cell, it's very crowded. You know, we think of a cell as maybe like a bag of molecules, but it's really densely packed in there. You know, there's not that much water. A lot of that water is bound to the molecules in there. It's more like a gel. So there's very high concentrations inside these cells. The interior is confined, and the fact that the molecules can kind of feel the edges of the boundary means that there's a tendency for the molecules to interact with each other. So maybe one data point that I'll just show here is that if you take an RNA that has to be collapsed onto itself in order to catalyze a reaction, and if it's in the kind of open configuration, it can't undergo reaction. And you have a version of that molecule which can't collapse, and so it is inactive. You can rescue that inactive molecule by putting it inside these protocells. So that's the kind of activity that you can get out of just putting something inside a container, not doing anything special to it except where it lives. Okay, so in the last 5 minutes, I just want to talk about a few applications of this type of work, you know, because I think when you do kind of curiosity-driven research rather than application-driven research, you always still have to justify, like, why are taxpayers spending money on this type of work? You know, why should we really care about this? So I'll give you just 3 little vignettes about how research on the origin of life has led to real-life applications. So one is the development of test tube evolution of RNA. So the ability to evolve RNA in the laboratory has led to, you know, discoveries about the origin of life, but it's also led to the potential to create RNA molecules that are also drugs. So if you select them in the right way, if you evolve them in the right way, you could make an RNA that maybe inhibits a protein. So here's one RNA that was evolved to inhibit a protein that's involved in macular degeneration. And this is, you know, maybe some of you have heard of it. It's one of the leading causes of blindness in older people. So here's, you know, a kind of application. in medicine. Here's an application in drug discovery where somebody, Dieter Brown's group, was interested in trying to mimic hydrothermal vents and cycling molecules through these systems where you had a high thermal gradient, like one side of this system was heated and the other side was cold, and trying to look at the convection cycles and what you could get out of that, what reactions you could drive prebiotically. His students realized that you could use this property, so they realized that different molecules move differently in this convection cell, and they realized that they could use these differences as an assay for biomolecular interactions. So now they have the spin-off company that uses this phenomenon and differences in the way that these molecules move for high throughput drug discovery. And this is also an application I love by Donna Blackman at Scripps Research in San Diego, where she was really interested in understanding what's called biological homochirality. So as you know, your hands, if you look at them, you've got a right hand and a left hand. They are very similar. They're mirror images of each other, but you cannot superimpose one on the other. And it turns out that biological molecules are only one hand if you extend that kind of structural property to molecules. Biological molecules are only one hand. So she was trying to understand how you get just one-handed molecules out of prebiotic synthesis. And she discovered several mechanisms to make this happen, and one of them turns out to be involved in like grinding crystals of glass together, which will kind of drive the solution toward one-handedness. And nowadays, that she was also a consultant for industry, for pharma industries, and nowadays those mechanisms that she discovered are being used for synthesis of molecules, so that you only get one-handed molecules which are bioactive. Okay. So with my last slide, I just want to give you a glimpse of where this kind of research is coming from. So it takes the people in the labs, academic labs. Here's my research group. Here we are kind of making the adenine structure on the floor there. And then, of course, it also takes people to be interested enough in this research to fund it, and that ultimately goes back to the American taxpayers who pay for the NSF, for NASA, and then also to private foundations who help us fill in the gap for these curiosity-driven projects as well. So thanks. I'll be happy to take questions after this.
Thank you very much, Irene. I'm going to pass some microphones around if you want to raise your hand if you have a question, and we'll be here for a little bit to take some questions. Ready?
Yeah, that's a great point. So the question is kind of like, how can we find evidence of evolving life? And that'll be really difficult, because then you would want to see like a progression of something older and something newer. So what I think people focus on is biosignatures, or what we think are like kind of correlates of life, or pointers toward life. So biosignatures being like liquid water. So some of the structures on Mars, I think you're talking about, they're pointing toward like liquid water. If you're thinking of like those Martian meteorites where it was thought to be structures similar to stromatolites, like microbial mats, there was, I think in the 1980s, a debate about whether some structures that were found on Martian meteorites were microbial fossils or were they made abiotically. And I think that the debate sort of settled, my understanding of it is that it sort of settled into the consensus being they were probably made inorganically, so not microbial fossils, but we would still love to see those. I think if you did find a microbial fossil, maybe that would tell us that there was some Darwinian evolution there, because there had to have been some progression from the molecules to something as complex as a microbe. But some of the things they're talking about, some biosignatures, I think are really more like signatures of potential for life, like liquid water. That's not really a sign of life, but it's a sign for the potential for life.
If I understood correctly, we took carbon, nitrogen, hydrogen, oxygen, and added heat or electricity, and then we created amino acids. And from there, we went to RNA, and from there, we came to the first living organs. And I know we have been able to replicate from molecules to amino acid in laboratory. Have we been able to take the next step in taking that into a living organism in the laboratory, or where we are?
Yeah, I think that where we are right now is the RNA replication is not quite efficient enough to make a copy of itself. So if you think about the Xerox machine kind of RNA, that RNA right now makes copies that are too fuzzy, too inaccurate to copy its own information. So it's not quite there yet. Having said that, it started out really poor, and it's been getting better and better. People have been engineering it, evolving it to get better and better over the past couple of decades. So I feel like that is a problem that can be solved in some years. So then we'll have a self-replicating RNA, I think. Once we have that, for trying to get that into cells, I think there are good model systems for making self-replicating vesicle membranes. So these like cell-like structures, they self-assemble because it's a thermodynamically downhill process. So they self-assemble. If you input a little bit of energy, it could just be like shaking or agitation. If you input some physical energy, that can break up these vesicles. So I think that could, that element, that ingredient could also be there pretty soon. One thing that we still have to work on is the interaction between this membrane and the RNAs. And I think we're still learning about what they do to each other. So I mentioned how like just being in this containment changes the properties of the RNA. I think we also want to understand how could the RNA maybe help grow the membrane. We've looked at some mechanisms there too. But understanding how these two elements interact with each other and can cooperate, I think will be really important. Once we get that sort of system working, then we also have to think about how did we get those RNA polymers from chemical replication, and can we do that in the lab? We can kind of write down what it looks like in theory, and we've got like bits and pieces of it, but everything needs to be like more efficient, more accurate, and things like that. So I'd say there's been progress in all of those areas, and toward making this RNA-based cell, like a very simple, and it's hard to call it life, but that's sort of what we're thinking in our mind is this RNA-based primitive cell that we really have to baby in the lab, but still would potentially be able to evolve. I feel like we are. I feel like there's a light at the end of the tunnel. I guess I would say.
Okay, we have one more question over here.
In high school, when dinosaurs were on there, I remember drawing the structure of a cell, and the endoplasmic reticulum was thought of as the home of RNA within the cell structure. Your current thinking suggests that now there are membranes around RNA itself. Is that changing the way we think of the cell, and can a cell just be RNA?
Yeah, that's a great question. So wonderful memory. I think what you're thinking of is the rough endoplasmic reticulum has all these dots on it, and those dots are ribosomes, and the ribosomes are mostly RNA, and a lot of the mass of a cell is in the ribosomes. So I think some of the where that kind of thinking is now, like where do we see these maybe primitive cell type organizations inside living cells? I think the closest analogy now that we're starting to learn about is RNA condensates. So it turns out that inside cells, and this was discovered maybe 10 or 15 years ago, that there are these like balls of RNA, highly dense, so mostly RNA, just a little bit of water in there, which is a separate phase from the cytoplasm. And those seem to be like regulatory bodies. They seem to have a lot of functions that we're still learning and understanding. And a condensate like that has very different properties than a RNA floating around in water. So one of the interesting properties people have seen is that some reactions go a lot more quickly inside those condensates. So maybe if you had a protocell, a simple cell that was really packed with RNA, that might give some advantages to that cell. So yeah, I think there's a lot to look forward to.
Private discussion later. Very interesting question and good answer. Okay, we have time for one or two more, and then we can hang around, but I want to keep everybody here for the after party. Okay.
Thank you very much. I happen to be in a social situation with many UCLA researchers in the biological sciences. Because of the withdrawal of funds, their forward progress had been stopped. Not all of them, but some of them. My question is, in your field, have you experienced diminished funds, and has it affected your forward progress?
Yes. So as some of you may know, as you know, more than $500 million of federal funding was frozen at UCLA. And the vast majority of that was NIH funding, National Institutes of Health funding. For my lab, that meant one of my grants, one NIH grant and also one NSF grant, National Science Foundation grant. So we did basically stop spending in significant ways during that time. It was very chaotic, honestly. As of 1 week ago, there was a ruling by a judge in a case that actually started back in February, which influenced us and resulted in a temporary injunction that caused the NIH to release those funds again. So we're trying to catch up now, and at the same time, having said that, some people were laid off, and there was kind of a general chaos. You can imagine just turning off the water in your house for 2 months. That was kind of what it was like. But we're all kind of trying to recover, and thank you for bringing that up.
Last question publicly, and then we can wrap it up.
I'm not sure this is a fair question, and maybe one of your other lectures will address it as cosmology, but given you start with one set of ingredients and you can have
That is a great question. I am agnostic but hopeful, I would say. I'm very hopeful.